Okay, for those who are joining in, I see our numbers are climbing here. We've got about almost 100 people. Just going to give it a minute for people to log in, and then we will begin. But as of now, we have 100 participants and climbing. All right. People are still coming in, but I'm just going to um, say hi to everyone and get this going. Welcome to Marine Biodiversity Networking Fridays. Um, it's exciting to have everyone here. My name is Neil Hammerschlag. I'm the executive director of the Shark Research Foundation. And I'm also an investigator with the Marine Biodiversity Observation Network. And I'm the lead of the BioTrack initiative, which is a collaborative project that uses animal telemetry to assess and monitor biodiversity hotspots where multiple marine species across Texas share habitat. So we're excited to have everyone here in the session. We're going to hear with updates and advances from the Animal Movement Biodiversity Observation Network, MoveBon, the Ocean Tracking Network, and the U.S. Animal Telemetry Network. Just to give you an idea of what's going to happen, we are going to have presentations from our speakers. Presentations will last a maximum of 40 minutes. After those presentations, we will have open discussion. So please put your questions in the Q&A, in the Q&A chat, and I will be pulling those questions for the speakers. So please hold your questions or put them in the Q&A chat for our speakers. But today, very excited to hear from Sarah Davidson, she's a researcher with the Max Planck Institute of Animal Behavior, and she's going to be talking about MoveBon. Jonathan Pye, who's the director of data operations at the Ocean Tracking Network. Megan McKinzie, who's the data manager for the Animal Telemetry Network with the Monterey Bay Aquarium Research Institute. And so without further ado, I'm going to hand it over to Sarah to kick us off.
All right, thanks, Neil. Let me just get sharing here. And all right, can you see my slides? All right, well, thank you everyone for being here today. Again, my name is Sarah Davidson. I'm the data curator for MoveBank, and I'm the interim co-chair of MoveBon. So I am excited to be here today presenting on behalf of my fellow co-chair, Lacey Huey, and the MoveBon leadership team. And I'll be introducing you to a new thematic biodiversity observation network for animal movement called MoveBon. And I'll tell you a bit about why and how we were created and what we're planning to do going forward. And then John and Meg will be describing some of their activities that are really well aligned with one of our first goals as a bond, which is to coordinate and grow efforts to mobilize existing movement data for policy impact. Across the globe, animals are constantly on the move. They're migrating, dispersing, and responding to changing environments. These movements shape ecosystems, signal environmental change, and connect disparate populations. So thanks to animal-borne sensors to monitor movement and behavior of these animals, we have collected billions of observations capturing these journeys in stunning detail. This theme of movement is broadly reflected in the language of many multilateral environmental agreements. So for example, movement is a key component of at least 9 of the Sustainable Development Goals, those SDGs you see up on the left, 15 targets of the Kunming-Montreal Global Biodiversity Framework, that's this GBF you see in the center, and they are themselves a central feature of the UN's Convention on Migratory Species. However, workflows to transform raw movement data into conservation policy remain underdeveloped. So one evidence of this is a recent systematic review of the Kunming-Montreal Global Biodiversity Framework, which found that in terrestrial realms, movement data was only used in one out of 30 GBF indicators. And so found that they have low relevance. But they noted specifically in the paper, it's not because they're not important, but because data are not available. Well, this is despite really large investments globally over decades in creating shared community platforms to discover, harmonize, and access these data. And so here, each dot you see is a whole data set. So some of these dots have millions of records behind them. And these are about 9,600 studies that are publicly discoverable online in one of these 8 big platforms. So that's really exciting, but what we see here is actually most of these data are not really that easy to access. So across all of these projects, only 12% offer public download. And so for most of the non-public projects, we're relying on like a tenuous connection to a single contact person to request access to use those data. So that really slows down things if you want to access things at a big scale. But we believe that the movement community is really primed for having global impact. We know there is a lot of data out there, and there are tools and networks to unlock it. And we need to just be able to scale all of these in order to leverage them for policy at a global scale. So we can point to existing successes. Policy relevant information derived from movement data are being made available to policymakers through a variety of dedicated efforts. So for example, tracking data having been incorporated into many migration atlases, including the UN CMS Atlas on Animal Migration. We have animal-borne ocean sensors, or ANIBOS, which is a developing network endorsed by UNESCO through the UN Ocean Decade to coordinate the integration of movement and environmental data from marine biologgers into the global ocean observing system. And we have BirdLife International's seabird tracking database, which has contributed to the designation of new marine protected areas. So we have the opportunity to dramatically scale these types of uses and impacts across species and regions, but we have some key challenges. So first, we have data fragmented, a lot of data hidden, and then limited interoperability between existing platforms. We have gaps, geographic and taxonomic gaps in data coverage. We have insufficient workflows or inefficient workflows for translating raw tracking data into policy relevant indicators. And then we also still have fundamental knowledge gaps in how do we scale from an individual to a community and ecosystem levels. And so the goals of MoveBon are to enhance science policy integration, so promoting a shared understanding of a shared science to policy pipeline for movement. And then we want to coordinate. So we want to collaboratively pursue opportunities within existing initiatives to close gaps in the information pipeline for movement data. We want to work directly with policymakers to develop indices that explicitly link changes in animal movement with biodiversity outcomes in ways that we can monitor over time. And we want to operationalize solutions, so by fostering relationships with signatory countries to develop frameworks, build capacity, and create national level case studies. So here is a quick look at the 2 years leading up to the creation, excuse me, of MoveBon. And so we started out with a bit of seed funding from the Smithsonian. And with that, we were able to attend a couple of global fora during 2024, and we hosted 2 workshops, one looking specifically at ungulate indicators and another one on really how are we going to form this bond. And we also ran a webinar series, which is still online at Wild Labs, to help kind of bring people together, give a little background information. We submitted an application this past summer and were endorsed by GeoBond in September. Here's a quick look at our leadership team. These are the leadership team that we're building out. What this is kind of formalized governance of the bond, and these are leaders of emerging working groups that kind of jumped in to start work on within the bond right away. And so here, just a quick plug, and we'll have another one at the end, but we really want to invite you to join us. We have, as of when I checked yesterday, 186 members, and we're definitely looking for more participation, in particular from people working in the marine domain. So we have the web, we'll post the link again as well, but yeah, please join us. And so what are the next steps coming up? So we'll be formalizing our working groups. We're working on publishing a few conceptual papers. We have actually right now, a bunch of our team is in Lima, where we are launching our first national case study at the International Conservation Technology Conference. So focusing on Peru, Peruvian stakeholders, data scientists, and people from the government all working together. And this will be our first time doing this. And then we'll be joining some global fora coming up this year. And what I want to talk about and what we'll be talking about in this session more is this step 5 here, which is this data pipeline development. So here is an envisioned workflow of our data policy pipeline. And the idea here is to scale the integration of animal movement into multilateral environmental agreements. And so that colored circle you see in the middle is showing, it's indicating the estimated technological readiness of movement data within this workflow. And so today, we're going to be focusing on this step here, where you see this is in yellow, and this indicates we have a little bit of a blocker to better integration with these multilateral agreements. And so we want to build on these existing successes with integrating data on community platforms by developing scalable pipelines to get more data into the hands of stakeholders and work with them to design policy relevant products. So a first step identified by MoveBond is to coordinate efforts to mobilize existing data from these community platforms, tracking data platforms, for example, by publishing it to the Global Biodiversity Information Facility, or GBIF, and the Ocean Biographic Information System, or OBIS. And these are key data platforms used for biodiversity research and policy. And so here I illustrate a prototype pipeline that takes data from MoveBank using an R package called MovePub, which is led by Peter Desmet at the Research Institute for Nature and Forest. This is under active use and development. And so through MoveBond, we're aiming to get input from policymakers and national efforts to maximize with use and usefulness of this and similar pipelines. Now, animal tracking through acoustic telemetry offers a really key opportunity for this, because when acoustic transmitters and receivers are owned by different parties across distributed networks, it is an absolute requirement that you're collaborating on shared databases to even get your data set. And so this really sets these infrastructures up to lead the rollout of operational pipelines to mobilize movement data. And so next, John Pye from the Ocean Tracking Network, and then followed by Meg McKinzie from the US Animal Tracking Network, will demonstrate how they are doing this and providing really a wonderful model that others can build on. All right, I'll pass it off to you, John.
Thanks so much, Sarah. This saves me a bit of time describing the natural advantage that acoustic telemetry has in bringing people together. I'm John Pye, Director of Data Operations for the Ocean Tracking Network. I am a computer scientist by trade, and what I have to do today is talk to you about biology, so please be gentle. I'm here to take you through how OTN is supporting policy-driven research across scales, and please do enjoy the pun. So we've defined acoustic telemetry. That is the central data set that OTN and its partners are curating. Tracking animals via these unique acoustic ping codes. First, you attach those ping codes to the animals that you're interested in. You deploy some listening stations underwater to record when those pingers are coming near. And then this high-level imagery compatibility that Sarah was talking about, that means every researcher who deploys receivers is extending that global observatory for you and everyone else. Each tagged animal then can be detected by everyone's field stations. So over the years, OTN-affiliated researchers have also attached listeners to autonomous surface vehicles, underwater profiling vehicles, and satellite-tracked animals. So for the past 5 years, our fleet of autonomous underwater vehicles have also been listening for and identifying whale calls as part of a multi-method monitoring program led by Transport Canada, which is intended to reduce ship strikes and entanglements in the seaways of the Gulf of St. Lawrence. I mention this because we are not tagging whales per se. They make their own noise, and we were able to listen for it. I want to be clear about that. We are designed at OTN to be a global research infrastructure network. So we're funded by the Canadian Foundation for Innovation. We're headquartered at Dalhousie University in Halifax, Nova Scotia. Adding value to aquatic animal tracking studies is a major pillar of the OTN mission. To do this, we provide researchers with listening instruments and we offer field support. But another big part of OTN's contribution is as digital data infrastructure through our data center. We've been around since 2008. We support Canadian and international research by deploying this communal listening equipment, training of new telemetry practitioners, and expanding partnerships to improve the capacity and the reach of the global network. The knowledge that is collected by OTN-affiliated researchers has been useful to policymakers, industry, Indigenous and coastal communities, and to the general public. Now, from the perspective of a data manager, and where my team's contribution is in research data management, quality control, and building and spreading standards, we have to think about acoustic telemetry data as the marriage of 3 data inputs, and humans are recording 2/3 of them by hand, which makes quality control really important. So acoustic receiver deployment history gives us the locations of detected tags. The tagging activity gives us the animal that was carrying the tag with the pin code at that time. And then the instrument records times and tag codes. You have to match these 3 bits of information together to give yourself this animal at this time in this place. The system that OTN has built and designed and continues to maintain ensures that every data contributor can be relied on to produce and share high-quality data. OTN's data system enforces standard formats, performs quality control tests, and executes a global-scale cross-matching and notification process, which allows all possible animal observations to be passed on to each of our affiliated researchers with confidence. In 2023, OTN's funding was fully renewed by the Canada Foundation for Innovation for another 6-year cycle. We will continue to operate through 2029 as a major science initiative. With this renewal came a new strategic plan, rededication to our core principles, track, connect, and transform, tracking and cataloging animal movement across broader geographic spaces and a wider range of taxa, connect animal tracking practitioners to one another, and mobilize the data and the knowledge they produce into spaces and formats that will guide policy, and help their work transform the way we conserve and sustain aquatic species. More recently, our funders have invited us to participate in shaping a new longer cycle program as one of the first major research facilities. This cycle would see OTN funded through 2040 and beyond. So data-wise, acoustic telemetry, OTN, we're a combination of many collaborators with each with their own individual questions. So what does collaboration with OTN look like for those individual researchers with those questions? I borrowed some slides from researchers at Dalhousie's Lennox Lab to help me illustrate how individual projects combine to create regional, national, and global scale biodiversity monitoring capacity. So I'm not a biologist. This is a short talk. We're going to skim. First, you have to know the mackerel fishery in Atlantic Canada. It's a bait fishery. It's currently under moratorium. Due to overexploitation, possibly climate change impacts, the spawning stock biomass of the northern contingent of Northwest Atlantic mackerel have been in the critical zone since 2011. DFO closed the commercial bait mackerel fisheries in 2022. This was a promising way to try to rebuild the population, but it revealed that there were numerous knowledge gaps about the species movements and biogeography that hinders the development of effective management that would benefit both the fishery and the mackerel population. So the Lennox Lab identified this deficit, and they secured funding to design and implement a telemetry study that leans on the existing infrastructure in the region to try and inform policymakers. And just some headlines from the local news that show that this is a big problem that we're all clued into here in Atlantic Canada, and we're interested in seeing this resolved. So the Lennox Lab were really interested in observing large-scale movements. They tagged 3 different populations of mackerel in different places around Atlantic Canada, but they only added new listening stations locally in Halifax's northwest arm. They were able to fill a gap in listening coverage, but depend on the listening efforts from OTN infrastructure, but also from all the other partner projects in the region, as well as those of our neighboring networks and the ACT and FACT network on the Atlantic North American coast. Thanks to the data that was returned from the network's many partners in Canada and the US, Morgan Pizak, who was a postdoc in the Lennox Lab at the time, was able to capture and quantify movement across management boundaries and national borders, identify between stock interactivity, and feed that information directly back to the policymakers who are making decisions that affect that stock. On the government research side here in Canada, there's a federal project that's using telemetry to understand species movement and space use and adapt policy. Analysis of this data is being undertaken by the Marine Conservation Research Group out of DFO Halifax. At DFO Halifax, they're evaluating marine protected areas in a variable by variable fashion, leveraging a combination of data sets, including their own animal tracking studies that they're submitting to OTN, but also other published tracking studies that other OTN partners have published to OBIS. to evaluate each metric of a given marine protected area and the richness of the data behind that evaluation. This group, I think, is the future of reproducible reporting and data-backed policy at DFO. They're active on GitHub. They're making reproducible workflows for these evaluations available to everyone. I have a great time following them on GitHub. I recommend it. So those are a couple of the partnerships that combine to make up a regional observatory in the Canadian Atlantic. On this map, these are the core OTN maintained lines in Atlantic Canada. Field technicians at OTN put these moorings out ourselves and we service them. But there have been hundreds of projects of varying scales and budgets, from those DFO maintained curtains in the Bay of Fundy, hello to Mark Trudell, and the Gulf of St. Lawrence, to other small-scale river projects across Atlantic Canada and New England that have contributed over the last 17 years to an understanding of animal movement in the Northwest Atlantic. Pretty incredible spatial and temporal coverage is achieved when you're able to put everybody together. We can also scale this up to network level coordination. So if we zoom out to the continent level, we're looking at 3 different major networks combining their efforts from the Arctic to the Caribbean. Projects dependent on this scale of collaboration were highlighted in a paper by Dr. Charles Bangley in 2020, and this map is an updated version of the one that was published in that paper. New compatibility coming online with Telemetry Network's iTag and RAFT will extend this coverage further through the US Gulf of Mexico and into the freshwater systems of the interior USA. The newest node out west, NPACT, will build on that fast-growing capacity of the existing OTN nodes and take it to the Pacific Basin. At that multi-network scale, we see numerous cases where this collaborative telemetry observing capacity is contributing data and analysis into policy, as with the CDAR assessments of multiple species populations, migration, space use, and health. These are out of the FACT and iTag networks. So now we zoom out to the global scale. We can imagine what is possible when all of us are building the same data set together. We connect telemetry observation networks in all parts of the world. We also lead the establishment and certification of standards and practices that can guarantee that accurate data gets in the hands of each participating researcher and published data set. OTN's data system has received Core Trust Seal certification. We've been accredited by the Intergovernmental Oceanographic Data Exchange Quality Management Framework, and recently we've been welcomed as a member of the World Data System. OTN also provides pipelines for all of our networks to publish their data upward through our connectivity to the Ocean Biodiversity Information System as a thematic node of the OBIS network, adding all of their knowledge to the global understanding of marine biodiversity. We're proudly representing Canada as a global leader in the best ways to track, connect, and transform this data into the places where it can have the greatest effect on policy, using OBIS as the springboard into Ocean Decade initiatives, proposed digital twins of the ocean, any number of regional to national level biodiversity knowledge products, and now to move on. We are also a participant in the emerging goose network called Anibos that Sarah mentioned. Tracking animals instrumented with satellite tags in real time is only the beginning, as they reach and can sample very difficult to reach parts of the global ocean and collect environmental data that is critical to physical modeling. So as a national data assembly center for the Anibos network, OTN intends to serve Canadian researchers who are instrumenting the animals with oceanographic sensors and help them to correct and connect the real-time data they're providing to the global telecommunications system. We recently completed a data pipeline pilot using Australian IMOS research program data that was in the Kerguelen Islands in Southern Ocean. Operational data streams that cover Canadian research are planned to be operational later this year. And so with Move On, Peter is in the audience as well, and I'm feeling good about showing off the title slide. We are participating in this exciting global scale exercise to get as much published animal movement data across domains into a thematic data portal backed by data sets registered in the Global Biodiversity Information Facility and the Ocean Biodiversity Information System. So across avian, marine, and terrestrial domains, we can describe the movements of individuals at a single standard, smoothing this pathway towards analysis and policy. At Living Data last year, this was Peter's title slide, challenging us all to curate and publish 1,000 biologging data sets, and OTN is excited to do our part in meeting that challenge. And that's it for me. Thanks for having me, and thanks for your interest in aquatic animal telemetry.
Thank you. Moving on to Meg.
Right. Hello, everyone. I am Megan McKinzie, and I am going to be giving you a brief introduction to the U.S. Animal Telemetry Network, focusing on some of our data archival pipelines and some of our recent efforts to mobilize data. As was already discussed and illustrated by Sarah and John, movement of marine animals are fundamental to the structure, connectivity, and function of ocean ecosystems, yet remain underrepresented in conservation, management, and policy frameworks. And again, this is part due to limited access to standardized, interoperable movement and environmental data. So today I'm going to be introducing you to some of the ways that the U.S. Animal Telemetry Network is attempting to open access to these valuable data streams. Now, the multi-agency U.S. Animal Telemetry Network was originally established in 2016. And as a network, we strive to provide unity, stability, and continuity to the national infrastructure that supports the collection, management, and availability of marine animal telemetry data and data products. And we also seek to promote the integration of these data into our national and global ocean observing system capabilities. And while ATN serves a broad community of scientists studying species ranging from small migratory fish to large marine mammals, so we are tagging whales, or at least our researchers are, across national and international waters, we are also working hard to support our stakeholders and data end users and the broader scientific community. And some of the ways that we are doing this is through helping to build and promote alliances and collaborations through community outreach and engagement, as well as through providing funding to help support baseline marine animal telemetry observations and research, and of course, through providing data management support services. Now, that is really the central mission of ATN's Data Assembly Center, which I help manage, or the DAC. Now, the DAC is a national level community resource that facilitates the aggregation of primarily satellite telemetry data from multiple sources and tag types. And we also provide data management tools and services to help support our researchers sort of through their entire data lifecycle. We also promote data standardization and help facilitate some QA/QC protocols of these data and metadata, as well as enable the display and access to these electronic tag data and data products, and provide resources that also help our researchers and data contributing members with permanent data archival at recognized data centers, which is really sort of the focus of today's talk. Now, as a quick side note, in addition to movement data, the DAC also disseminates near real-time ocean profile data collected by animal-borne sensor tags and integrates this information into Anibas. Now, these observations, including temperature and salinity profiles, are made available for assimilation into regional and global weather and climate forecasts and models, significantly enhancing ocean observing capacity, particularly in data-sparse regions, and further strengthening the links between biological and physical oceanography data. Now, it's complementary to these efforts are our data archival pipelines, particularly in light of what John just shared about opening up some real-time pipelines to serve these data out to repositories like OBIS. But we're going to be focusing on more of a delayed mode product. So after deployments are complete, some of our data pipelines to, again, mobilize those completed data sets. So the ATN currently supports the public dissemination of telemetry data at the Data Observation Network for Earth, Data 1, and the permanent archiving of satellite telemetry data sets at NOAA's National Center for Environmental Information, or NCEI, and then the subsequent transformation of these data sets into Darwin Core compliant products for the publication through the OBIS Biodiversity Information System, again, or OBIS, and the Global Biodiversity Information Facility, or GBIF. So we're going to dive into that process in a bit more detail. This is going to get a little in the weeds, so just bear with me for a few quick moments. All right. So focusing on our pipelines again to serve data to NCEI and then ultimately onto OBIS and GBIF. So as I mentioned earlier, the DAC is primarily ingesting satellite telemetry data. And most of this data is coming from Argos-based platforms. So these are things like your Spot, Splash, satellite relay data loggers, your CTT type tags, and of course your PAT tags. But again, we're going to be focusing on these sort of former suite of near real-time transmitting tags. So ATN satellite telemetry data can either be auto-ingested directly from a tag manufacturer or from the CLS's Woods Hole group via an API, an FTP, or a WAF, or it can be manually uploaded into our system via one of our online platforms. And data are ingested usually in tabular format as a text or CSV file, either again near real-time or in delayed mode after deployment is complete. Now, these data files, of course, are going to vary from manufacturer to manufacturer and tag type to tag type, but typically include things like relevant IDs, type date stamps, lat longs, location classes, and associated air parameters such as air radius, and if applicable, any depth or sensory information. Now, as John sort of alluded to earlier, we also collect robust metadata at 3 different levels: project, deployment, and data sets. And these information is provided directly, again, by our PIs through one of our online platforms. So just briefly, project metadata is including things like your title, abstract purpose, the creator and contributor contact details and roles, while deployment metadata are capturing things like your tag manufacturer, tag model, associated IDs for your serial PTT number, and then, of course, information about the tag animal, its common scientific name, API ID, unique animal IDs. Then, of course, also that suite of life history and morphometric information, such as life stage, age, class, length, weight, sex, and then information about the deployment itself, its when and its where. And then the data set level metadata goes into even more details about the when, where, why, whom, and by how these data were collected and transformed. So once we have the raw data files and the corresponding metadata, these auto-ingested data are run through IUS-QC protocols. So they are run through what's called QuarTog, which really just provides very sort of high-level whole ocean basin sanity checks. So this is kind of a one-size-fits-all approach. Flags are then embedded in the data, but no data are thrown out. From there, the raw manufacturer files are converted into an intermediate Parquet file. And then once the deployments are complete and the data is out of embargo, these intermediate QC files are then merged with the provided metadata and converted into standardized netcdf4 files. And while I don't have time to go into the full file specification, I do want to highlight just a few key components. So these files do follow climate and forecast, attribute convention for data discovery, as well as IUS, NCEI, and ATN, or really community-specific conventions. They have definable attributes and units. We utilize a variety of NERC vocabularies with things like creator and contributor roles, platform and instrument type, as well as sex, life stage, and links. And of course, we're utilizing some specific GCMD keyword libraries. And this template has been approved by the NCEI Dataset Readiness Review Board and is available on the IUS website and integrated into our compliance checkers. So I encourage anybody who's interested to go to our website to review the full specification. And of course, you can comment or contribute to discussion about these templates within our GitHub. All right, so moving on. So these standardized netcdf files are then served directly to NCEI via WAF as individual deployment files. Once at NCEI, they are grouped by institution and archived under the ATN collection. And the corresponding DOI references the collection as a whole, while individual deployments are referenced by unique accessor numbers. So as long as we have the auto-ingested data files and full suite of required metadata, this is more or less an automated process that requires very little effort by our contributing PIs. All right, moving on. So from there, data are harvested directly from the parent directory at NCEI and then converted into Darwin Core archives using the ATN to OVIS package that was developed by Matt Biddle, the IUS DMAT community manager. So essentially, the data are read directly from the netcdf files and used to populate the standard fields of the Darwin Core occurrence records. Data are filtered to acceptable locations by removing low-quality positions, in this case, location classes A, B, and Z, and soon positions that fail or are flagged as suspect during the previously mentioned QC procedures. And then the remaining positions are subsetted to the first detection per hour. The package also reads in the metadata directly from the netcdf files and uses it to populate the standard fields of the event core records and EMOF record, including information on the animal tag and relevant IDs. It also creates robust EML metadata records and the XML file for additional information that's contained within the netcdf files, as well as some information that's harvested from the NCI metadata record directly. And of course, we pay close attention to make sure we're including relevant data links, references, creator and contributor details, and citations to other versions of these data that may be contained in other repositories. And then finally, all this information is zipped together as a Darwin Core archive and automatically published to OBIS USA Integrated Publishing Toolkit, where they are then linked to the ATN collection and on to GBIF and other initiatives such as biologging. Now, this ATN to OBIS package and all of its automated processes, including the use of the IPT, heavily leverages on what was designed and built by the Ocean Tracking Network for serving acoustic telemetry data to OBIS and beyond. So there are a lot of similarities in how we subset the data and, of course, in the file structures themselves. And this really enables ATN to also participate in the MoveOn effort that John mentioned earlier, with the goal of publishing as much animal movement data as possible from cross domains and taxa into a single thematic portal. And before I wrap up, I just want to quickly highlight a multinational, multi-network success story, Operational BioTrack. Now, BioTrack really embodies what can be accomplished by leveraging network resources from ATN, OTN, and their regional affiliated networks, as well as their developed technologies and data pipelines. And through the utilization of contributed data, along with standardized data outputs for public endpoints like OBIS and GBIF, Dr. Hammerschlag and his team are working to identify and characterize shared multi-species hotspots, determine how these hotspots overlap with place-based management and resource use areas, and are even working towards predicting how future ocean warming may impact the location, timing, and makeup of these hotspots, working towards developing mechanisms to identify signals of change. And together, the initiatives, networks, and contributed data pipelines we've highlighted today allow us to aggregate data across domain and taxa, integrate it into a single thematic data portal, such as Wild Moves, and standardize it into a united public-facing data product, as illustrated here with the Wild Moves occurrence maps. And these efforts really elevate marine animal telemetry data from individual research outputs to globally discoverable biodiversity and ocean observing resources, enhancing their long-term value, accessibility, and relevance for ecosystem science, climate research, and evidence-based marine policy, as well as management and conservation frameworks. And with that, I will end with the ATN seal of approval and a call to action. We cannot do any of this without you, so please get involved. Register with MoveOn, participate in one of its many committees and working groups, contribute to your regional, national, and global networks, and integrate your data today. It's time to join the movement. Now, first, if you have any questions, please don't hesitate to get in touch, and we're looking forward to chatting with you now. Thanks.
All right. Thank you so much. Great presentations, really informative. You got really good quality, so thank you. So now we're going to open it up to questions and from people. Please put it in the Q&A. And in addition to that, obviously our speakers can ask questions of one another or the audience as well. So we're gonna kick it off. I guess we'll still wait for some questions, but there was one that showed up, and there seems to be one of the questions comes from George asking, are there any eDNA factors in all of this data collecting happening?
Yeah, Sarah, do you want to go first? I know some things, but
Okay, well, I mean, so certainly there's eDNA adjacent to stuff we've been talking about. And we focused today on like the collection of kind of individual type movement data. But certainly like in the context of for example, like GBIF and OBIS, those are places where eDNA data can be hosted right alongside movement data. The platforms we work on, as far as, you guys can answer too, but like MoveBank doesn't store eDNA data, for example. But yeah, so this is kind of why like putting them all in a place like GBIF and OBIS is a really good way to kind of get at somebody who doesn't really care how we got the information, but they want the information, right? Let's put it in one place so that people can really take advantage of all of that. Yeah.
Yeah, that's exactly what I was thinking as well, Sarah, that there are definitely other efforts to collect animal presence data using eDNA. I linked in the chat to one that I'm aware of that's feeding to OBIS, that OBIS is looking for more data contributors for. But that is somewhat outside of our own scope, but will be sitting right next to our data in the big global databases.
And
yeah, and I don't really have much. Oh, sorry, go ahead, Neil.
No, go ahead. Go ahead and add.
I was just going to say that ATN sits under the Integrated Ocean Observing System umbrella and is part of this larger Unified Marine Life program. And part of that program is focusing on collecting eDNA data along with other sort of HAB-related information. And again, there's a lot of analogous pipelines that allow us to integrate these data together, as Sarah and John mentioned. Go ahead, Neil.
Yeah, and one things of the BioTrack initiative that Megan spoke about, which I run, we are interested in how the two types of information can inform one another or not. And so we are actually we have a working group meeting. It's in anyone's interested. Coming up early next month with the goal of with people who have eDNA data, particularly right now, we're going to focus on the East Coast of the US. Anyone who's got data, tracking data, or eDNA data, we're going to try to compare and contrast information gathered. And so that meeting is going to try to identify who people have data sets, but also maybe approach analytical approaches, because also eDNA and tracking data is captured at different levels, different taxonomic resolutions, and to kind of come up with some valuable questions about how the one can help maybe inform the other, or how they're related to one another, patterns in biodiversity at the eDNA level and at the animal tracking level, and maybe some key questions. So anyone who's interested in that discussion, maybe shoot me an email and I'll send you an invite. I'll put my email in the chat and we'll invite you to that working group if anyone is interested in kind of the combo of eDNA and tracking, and how they can maybe help inform one another, or at least look for patterns in biodiversity using both. So we do have some more questions in the chat, and some of these questions, I would say maybe people coming in at different times, but maybe they were somewhat covered in the talk. But I guess one of the questions is, are there plans to integrate movement and telemetry data sets from platforms like OTN, ATN, or OBIS to support long-term population monitoring under climate change and environmental change?
Because OTN's first on the list, I'll say that that's part of the reason that we're publishing this data to OBIS. The intention is that it is there, data from as far back as 2005 is in OBIS and is there to be picked up to create these kind of longitudinal analyses. There are groups that have been using OBIS this way, not necessarily just for tracking data, but for generally what's in OBIS. There are drawbacks to using OBIS as a kind of a catalog of the places that we're currently sampling. Initiatives like animal tracking help to offset some of the human-centric sort of focus that these databases can have. But that is kind of why we contribute to the broader database. We intend for it to be taken up not just by people looking for animal tracking data, but also looking for all the different ways of having animal presence data recorded.
I can add, so this question, I think, overlaps a bit with this other question of how wild moves is different from MoveBank. So maybe I can answer these kind of together. So I showed you that map, that global map with all those dots from different platforms, right? So there was MoveBank, there was OTN, there's ATN, there were others as well. And if I want movement data, the first thing well, can I just go to one place and find all of them? Can I click on that map and get data from all of you? And that has always been a question. And so the question has been, well, if people are using Ocean Tracking Network, should they also put the data in MoveBank? And so we, Meg and John, we've heard this, for like over a decade, this is like comes up. And so enter wild move. So the idea is, let's all, because we're all doing kind of detailed things for different user groups, could we all have these pipelines to put the data in GBIF and OBIS? And what wild moves is essentially just a window into GBIF to take anybody who wants to publish tracking data. They can follow a few things in how they create their Darwin Core archives, which is how the data format that is understood and received by GBIF. You publish it there, and if you've put the right little flags in, it will show up in this Wild Moves portal. And so that is the whole concept behind that, is that we could all take our data and make a representation of it as biodiversity data on GBIF and OBIS. And so that's where that Wild Moves really comes in there, is that we're hoping will become the place that is this discovery portal that reaches across different databases. Specifically for MoveBank, I can say that, like, for example, most of the data on MoveBank are not animal tracking data. There are other sensor data that are also collected by these tags. And when you get into that level of detail, it gets really complicated. It becomes a blocker to putting the data in GBIF. GBIF isn't set up for doing the kind of stuff that MoveBank is for researchers. And so instead of trying to make one solution that fits everything. We can take a representation of a data set on MoveBank and put it in GBIF in kind of the language that the people going there can understand. And then each one of the data sets that are on GBIF or OBIS has a data set, and you can link back to, hey, this is from OTN. You want all the, you want all of the receiver pings. You go back to OTN and you talk to them about it. Or you go back to MoveBank if you want all the gigabytes of accelerometry that went along with this track. And so that's, yes. So I hope that helps maybe explain where these pieces fit together.
Yeah, I think that was really well said, Sarah. And, ATN handles this sort of in a similar way. And again, there's, of course, the Darwin core versions of these data that are served to OBIS and GBIF. But if you're really trying to dial down in to the raw data streams, we are serving those data to places like NCEI, or in some cases to Data One. And so you can get different flavors of the data that are meeting different needs for different members of the community or end users or stakeholders. And so you need to go to the sort of respective repositories to pull the sort of subset of the data that you may be most interested in. And again, we tried really hard, I think all of us, to ensure that those connections and links are maintained through those various aspects of the data. And so they sort of cross-reference each other. So you know where to go to find the raw data or to go to find the sort of Darwin core versions of the data. And so it's easily accessible and it tracks sort of the provenance of the data from collection through mobilization. And really ensures that we're giving appropriate recognition to the researchers, the individuals, the institutions that are collecting this information on everybody's behalf.
Yeah, that is such an interesting and challenging aspect of publishing acoustic telemetry data, as there could be 20 different investigators contributing to each individual tagging program, and you don't know who they are until they start seeing your fish. So the system that OTN set up had to account for a growing set of collaborators across different editions of individual data sets that are built and published, and it works very well. The citations are on a receiver by receiver basis in the data set. You can see who is contributing what, and they're all listed as data contributors in the archives, which is great. And we're working with NCEI, following the path that ATN took, to try to preserve those myriad contributors to each of these data sets as we're archiving them. And it's looking good so far for us, but we have an interesting use case where you may already be a collaborator on a tracking project and not know it yet.
Yeah, certainly some unique challenges in the acoustic telemetry realm, just due to the large volume of collaboration and contributors that make up individual data sets.
So let me ask you a question, because I think, maybe it's not clear to people. Okay, so let's say someone is, first of all, the Wild Labs data are data from all the networks that are published on OBIS and GBIF. That is correct?
Yeah, let me just add though that it is really brand new. We couldn't show you that link like 3 weeks ago or something. It was really new. So you are not going to find even 1% of what's out there, but we're just starting it now. But yes, that's the idea.
Yeah, but it's only those that are already published publicly to OBIS and GBIF. Yeah, so that's an important difference when trying to, one of the questions, how is MoveBank different from Wild Labs? Well, MoveBank has data that shows data that's being reposited. That's not just, maybe you can explain how they're, how is OTN different from Wild Labs? How is what MoveBank does different from Wild Labs?
Yeah, I think a piece there is that, so everything on GBIF and OBIS is public. There's kind of there's no embargo. It's just public if it's there. Whereas I think all of our platforms have some degree of it doesn't have to be public. We're collecting real-time, really real-time data of endangered species, right? So we can't have a public default. Even a public, you can look at it default, And so it is this constant, well, I would like to do, let's see the question, can we do an integrated analysis of all the data already available? Well, most people aren't just going to let anybody take their data to do an integrated analysis of all the data that are available, right? So there's a lot of give and take of people who are like, hey, I'm just starting my PhD. I'm just putting a few tags on. No, they don't want it to be made public right away, And so there's all kinds of reasons why it isn't all public. And so that's a lot of what we're working with. And the nice thing about GBIF also is that that's a space, and OBIS is a space where the version that we can make publicly, we can put here. And one thing I think is exciting in my perspective, is that it also makes this idea that we're not sharing everything. It gives us a space where, like, could those R packages, right, that we're making, could you say, listen, I'll share like a monthly occurrence for my data set. That's no problem. That's not overlapping with our research goals, and it's not going to be a threat to the animals, right? Could I publish that to GBIF, and then people can find me, and if they really want the 1 second resolution, they can contact me for it. So I think it also creates a space for some like hybrid solutions. We can make the data, we can expose some of the data without exposing everything. So I don't know if that maybe helps.
And I'd also like to make a point here about the value of these besides, making data available publicly. They also keep data archived privately and as well as publicly. So it's a way to safeguard data. So it's not just sitting on someone's hard drive that, something happens to that person, it disappears. So it's sitting somewhere, it's in a safe that is being locked away and taken very good care of. And that's also particularly important when it comes to some publications now are requiring, a lot of publications requiring that data be archived in a public repository. So this is a way you can meet those needs as well as the safeguarding of the data, but also it be available in an archive, whether it's public or private, that means, that satisfies those journal publication requirements.
Yeah, that's a great point, Neil. And because like all of these projects on the acoustic tracking side are intercollaborating, it's so important to curate immediately and share with researchers immediately, and then have that pathway to publication existing on the day that these data sets can move, but have the best possible data in every repository as soon as we can from all these contributing projects. So we had this pressure at OTN to make sure that what we're sharing immediately with researchers is as good as we can make it. And then that results in each of our networks having the best possible snapshot of a project at any given time from where you can publish it with confidence. So we took the requirement set of acoustic telemetry and turned it to our advantage when creating publication endpoints, like at the individual network level.
We have a question, a great question from Connie, and she wonders, these available data sets that are found on these different networks, is it when you go into when you go into your websites or your portals, is it easy to filter and know which ones are, essentially public and accessible and which ones aren't? Is there, basically, is it very transparent which ones are available publicly, and to filter and search for those?
Yeah, you can filter for them, and then of course if you try and download data for something that's not available to you, you will just say no data available for download. You will see. So like on MoveBank, all the projects have a colored dot, and the colored dot, depending on the color, I can view a summary, a written summary, I can look at the tracks, or I can download the data. Those are 3 categories. And you can also, so you can visually see that, and then you can filter by that, for example. Also using the API, you can do the same.
And ATN is similar. We do also have our own public-facing data portal where the data that is integrated into ATN has been visualized and some sort of high-level contact information should you want to reach out. And sort of within the data catalog that is feeding the visualizations, there are sort of flags that indicate which data sets are archived and therefore have public data available. And if you see those archive flags, and then there's some sort of additional access tabs within each of the individual projects that allow you to get to sort of the citations and to link out to the public available archives, as well as to download the data directly from the data portal. It is sometimes a little tricky to navigate all those aspects of the system if you aren't familiar with it, but they are there. And then, of course, these data are kind of then pulled together at these various collections, whether it's the ATN collection at NCAI or the ATN collection at OBIS. And so there is sort of ways that you can see the subsets of the data that are public there. And I'll just say that presently for ATN, about 25% of all the data we have integrated into our network is currently fully public.
We have not too much more time, but there's been a few questions about the lack of representation of the global South. and would like to hear about from you guys about how we can fill those gaps, and what are your groups doing to help fill those gaps.
Yeah, Zeka Milbert is one of the ones asking the questions. Hi to Zeka. We did have an attempt there at a node in Brazil, and we'd be eager to try to rekindle that. And I think we're working behind the scenes with him and some others to do that. But on my side, it's about having local folks who are interested in aggregating, and that's the first ingredient of building up a regional network.
Yeah, go ahead, Sarah.
A couple things I can add is, so one is that the data privacy becomes a big issue there too. So people not necessarily wanting to make their data available to, other institutions they don't know, people they're not connected with, without, so there's a trust building, and there's are the tools helpful for you? There are, I mean, at least in the terrestrial realm, there are a lot of wildlife tracking data collected in the global south. They can be really, really sensitive because here we have poaching of like critically endangered species. So their priority is to keep these animals alive. It's not to often, okay, so I'm generalizing, but often the priority is wildlife management in real time, and less so, an academic exercise or aggregation of 20 years of data or something. So I would say there, AFREMOVE, this is also in the terrestrial realm, is a specifically focused on doing pulling together data and working together on collaborative research work within Africa. And I also then, my second point I want to highlight is that MoveBond, I mean, kind of one of the things about MoveBond, if you look at if you're trying to maintain global biodiversity, then you're going to go to the places where you need data, and you're going to organize there rather than where the resources happen to be. So it gives us this wonderful incentive to try and like really align around gaps in a way that an individual research group, well, they have the money to do from the government that wants them to study this, right? And so MoveBond and working with these multilateral environmental agreements, I think gives us a new way to connect around spaces where we're missing data, and really do kind of some dedicated work to build that out. So that is actually, exactly one of the exciting things about MoveBond, I think.
Yeah, and getting sort of boots on the ground in the area to connect with these local researchers so they can understand what we're doing and why we're doing it, I think is really going to help to mobilize some of those data. And that's, part of this case study effort that's happening in Peru right now is, again, to help work with those local communities to mobilize some of this information from typically data sparse regions.
Thanks. Coming into our last minute here, Elizabeth asked, wondering whether Wild Moves will be integrating the Goose bioecom metadata portal, or vice versa, because that would be a great resource for research focused on essential ocean variables.
So my understanding of the Goose bioeco portal, we put all of our listening efforts on there at OTN. It's basically an endpoint that can harvest in the same way that Wild Moves is the set of things that we're currently publishing. So I think that's the way to think of it, as a data set and then two endpoints that can serve what we're finding at the data set level. So GBIF and OBIS is the data set, Goose bioeco as a harvester of that data. That's a way we should set it up.
Okay, great. Well, I want to thank our panel for great presentations. I would love to thank everyone who tuned in. At point we had, I think, close to 160 people, maybe more, and great questions coming in. So thank you everybody for just a great effort and to their interest and contributions to animal tracking observations and using it to support policy and information to the public.
Yes, thank you so much for having
Thanks,
us. This has been wonderful.
Thanks, Neil.
Thanks for the opportunity. Thanks, Neil.
Thank you. Thank you very much. It was a great session. We still have some people here.